Okay, I think we're recording now. So welcome everybody. I seem to be losing my voice today too, so I'm not going to talk very much. But this is the very first meeting of our full group of the Zfish project. And I think we've got pretty much everybody who's here part of the group right now. And what I want to do, again, just to start off, is to do introductions. If we can go around. And at the table there, could we start with Piyamika when we get there? And then maybe you can pass it around the table, but we'll do you last, if that's okay. So as you all know, I'm Amelia Martins, and I'm a co-PI, co-director of this project. And I see my role as really being on the outside. I'm trying to be the person who coordinates the big picture across all the smaller project pieces, and who does things like sets up this meeting. Let me pass it to, actually, let's just get Piyamika and not the rest of the table. And then we'll come back here, yeah.
Hello, my name is Piyamika Suryampala. Hi Monse. I am the co-PI of this project working with Emilia. And one thing that Emilia didn't mention, that she basically managed the budget, which I'm pretty bad at. Like, I'm not good with numbers, especially when there's a dollar sign in front of it. So she takes care of it. But in terms of the project, yeah, I was involved with recruiting students and looking for postdocs. But my main role is to run and coordinate the experiments that we are doing in the lab. So it's kind of like the data collection, maintaining the zebrafish lab, and communicating with the Arizona game and fish people so that we can complete that piece of the project. So kind of like the on-ground person of the project. Yeah, I think that's me.
Thank you. Now, if we could get the people who are on Zoom, like whatever order you want to do it in.
Are we doing popcorn?
Sure.
So my name is DeLawrence Sykes. I'm an assistant professor at Berry College. My role as a part of the project is to do some cellular molecular work with respect to how the environment influences TMBS-induced colitis in the fish. So we're going to induce colitis in the fish and see how previous experience in their habitats influences their colitis progression.
What is colitis in fish?
It's inflammation of their GI tract. That's
what I was thinking, but okay, cool. I missed that on here.
It's in there somewhere, I think.
Yeah, no, I don't think it's not, but I must not. It's been a while since I've looked at it too.
Yeah, actually, it's not in the proposal, right? So what?
Oh, good. I feel bad.
It's an add-on. So DeLawrence is in the proposal because we were doing enrichment, right? But he's been working on colitis lately, and we thought it was a great opportunity to try something else. So we're just doing a little tiny touch of that. Christina, maybe?
Sure. Hi, everyone. So my name is Christina. I'm currently a postdoc at the Cavanilles Institute of Biodiversity and Evolutionary Biology that is in Valencia, Spain, but I will be very soon joining the lab yet again. So my second time in Arizona, and I will be joining AIM. I think it's AIM 1.4. It's the AIM about gene expression. So I'll be working on some transcriptomics with zebrafish after they have been exposed to different treatments to see how the environment affects gene expression, basically. So we did something similar with lizards while I was over there. So the idea is to try to do something like that. So I hope I can be of help with that.
Monse? Or Caitlin, however you want to do it.
Okay, I'll go since I was muted. So I'm involved in measuring reproductive hormones associated with stress resilience behavior of fish to, I guess, cognition and what was the other assay? well, behavioral and cognitive assays and in relation to also enrichment. And I've been collaborating on this sort of thing with Emilia and Montse already. And mostly so far, just Paminka's been, we just sort of discussed how her setup's going to look like so that when it's time to do the hormones, they can get them easily, and then I'll work on the rest of the data from there, extractions.
Thank you. We should mention that you're in Texas too.
Okay, so yeah, so I'm at Texas State University, professor of biology, and I was just in Spain for 3 months, also in Oviedo, for my sabbatical up at the Biodiversity Institute near Oviedo with a collaborator working on hormones.
Monse, I think you're up.
I'm sorry. My connection is not very good. I'm sorry. I am Montserrat. I am in the National University at Mexico City. I am an associate professor, and I did a postdoc with Emilia during the pandemic. And then I went with Caitlin for a little while. And I think I am joining the part with the hormones also, working with Caitlin. My main research goes also towards that same theme, that is mainly I am an ecophysiologist now, but a behavioral ecophysiologist. So I really enjoy asking questions about how animals respond to different types of disturbance. With Emilia, I actually work with BPA and also just the physical presence of plastics. And now in my lab, I am working with development and how development responds to different types of pollution, like chemical pollution, but also sensory pollution, like light at night and noise pollution. And yeah, I think that's it.
Thank you. And Vincent, are you there? Yeah.
Yeah, can you hear me? Okay, my name is Vincent. I'm currently a sophomore at Berry College, and I'll be helping Dr. Syke with his work.
Wonderful. Thank you. Can we go to the main lab now and maybe just pass around the microphone? Gons, you could start us.
I start? Okay, my name is Gonzalo Ramirez. I am a postdoc at the Martins Lab. Currently, I'm working on analyzing sexual dimorphism of the skulls of lizards. So I know very little about fish, but this will be very interesting.
I'm Tony. I'm a second year PhD student in the lab. I'm also studying the Scaloperus lizards, looking at the jaw morphology. Yeah.
That's Tony Mesa.
I'm Sean Snyder. I'm in Dr. Sariampola's lab. I study, the enrichments and its effect on exposure to novel objects specifically, and also a little bit of the Allen exposure with the boldness assay. I'm currently a junior. I'm in my undergraduate here at ASU. So.
Hello, I'm Hope Abercrombie. I'm also on the lizard side of things. I'm just studying how they interact with the habitat.
Hello, I'm Melissa Ian. I'm a first-year PhD student here in the Martins Lab, and I am taking on part of the Rio, Texas project with the zebrafish, and also doing a study comparing native dace with the invasive dace here.
Hello, my name is Kimberly Lapidario. I am a first-year PhD student in the Martins Lab on the zebrafish side. I was working this summer on the experiment, and I'm really interested in cognition and stress in relation to cognition, as well as enrichment in relation to cognition.
Hello, I'm Vaishnavi Chinta, and I'm a senior undergraduate here at the Martins Lab, and I'm also working with zebrafish specifically, but I'm looking into the OMR aspect of it, so the optomotor response and how enrichment and Alan have affected that.
Hello, I'm Teresa Nguyen, and I'm doing my thesis on the transcriptome project as well on zebrafish.
Hi, I'm Manasa Sriram. I'm a third-year undergraduate in the Martins Lab, and I'm also pursuing a thesis conjointly with Vaishnavi, and my focus is more so on enrichment in ALAN and the impacts of that on visual acuity.
Is that all the way around? So that's us. It's a pretty cool group, I think. I think we've amassed an interesting, diverse combination of people. So what we have for the second part of this is really just to go through some of the results from the experiment that that lots of people at this point have been participating in over last summer. So I have the slides from everybody, and I will share to get that up. And I think we're going to start with Piyamika giving us a quick summary of what was actually done. And then we just wanted to share some preliminary results and get some input on what has been found so far. Can you guys see my screen?
Yes, we can. Huh? I don't know. It's from here and there. All right, so Amelia, first slide. Back. All right, so the plan for today is to tell you about the summer of 24 in Martin's lab and a little bit about the grant proposal that we were working on. The next one, Amelia. All right, so I want to tell you a little bit about, a little bit about the project that we are working on. In 2023, in October, I think, we got funded by the National Science Foundation to continue our research on sensory resilience to climate change. And this is a proposal that Emilia have been working on for like 2 years. And the first round, it got rejected, but the second round, we got lucky. And that's why we are all here today talking about this project. So in this particular project, we really want to talk about like how animals rely on sensory modalities to keep up with the environmental change, mainly because that sensory systems are at the forefront of detecting environmental change. And it's kind of like the fundamental link that will connect an organism's environment to the responses that they display. So it's really important that we understand how they use these sensory mechanisms to keep up with the environmental change. I think the next slide. Okay, so this is kind of like the overarching goal of our entire proposal. So we have 3 main aims. The first aim is to look at how animals rely on sensory systems to keep up with environmental change. So we are going to be looking at things like, are they displaying shift between modalities so that they can continue to maintain their general behavior? Or are they completely losing the ability to display, maintain their general behavior because the environmental fluctuations are too rapid for them to keep up with it? And the different mechanisms or different climate factors that we will be looking at are the increase in temperature, acidification, flow fluctuations, and exposure to artificial light at night, ALAN. Exposure to ALAN is not really a climate threat or a climate factor, but it's one of the very common environmental alteration that many organisms experience, especially zebrafish, because they live in environments very close to habitats that's occupied by humans. So because of the rapid growth of human population and urbanization, exposure to LN is becoming a really major concern related to animals and their survival. And in this aim-1, we will also be looking at, in addition to behavioral and physiological sensory mechanisms, we'll also be looking at transcriptomes, how they are expressed in response to these different climate factors. And this is where Christina's expertise will come in. And in the aim-2, we are going to be looking at what are the strategies that we can use to improve the resilience of these organisms, specifically focusing on sensory experiences. And we will be using hormone physiology and their previous sensory experiences as two possible strategies that we can enhance the resilience to environmental change. And this is what we were working on past summer, specifically focusing on sensory experience. Can we use sensory experience as a strategy to enhance resilience of zebrafish? And aim-3 is where we will combine all the knowledge that we gather from aim-1 and aim-2 to understand whether we can actually use this knowledge to enhance resilience and persistence of Arizona desert fish. So this is kind of like what we are doing just to make sure that everyone's on the same page in terms of our grant proposal. Yeah, so summer 24 is mainly focused on using sensory experience as a potential strategy to enhance resilience to climate change. And we specifically wanted to focus on sensory experience because with the previous work, we know that the current or the recent experiences in social and physical environments have direct implications for the responses, behavioral, physiological responses that animals display. For example, we know that young trout, when they are kept in low density environments, they tend to have higher survival rate, which is really important information if you're thinking about how we can enhance the survival rate of like hatchery raised individuals. When we introduce them into their native habitats, will they be doing well? And also in male guppies, we know if they are kept in only male majority tanks, they tend to display like a weak mating effort, so which can be affecting their mating behavior. So these are the first two examples that I have kind of talks about how their social environment, social context, or the conspecific that they're interacting with are going to be affecting their responses, behavioral or physiological responses. And then we know a whole lot about how the experiences in different physical environments can also influence the responses of organisms. With the work that DeLawrence have done, we know that zebrafish, when we keep them in physically enriched environments, they tend to form very tightly knit shoals. And it's really important for social animals because tightly knit shoals provide multiple different benefits, like evading predation, like reducing the risk of predation, things like that. And also, we know that young fish that are kept in simple physical environments with very little structural complexity, they tend to have a weak response to spatial learning, and also they tend to have smaller brains, which is really important. Like cognitive capabilities are really important if you are trying to navigate new environments. Like when you're thinking about environmental perturbations, your environment is changing rapidly, but if you are not capable, if your cognition is not capable of keeping up with these rapid changes, these individuals might not do so well with the changes in the rapid changes in their habitats. Next slide. So in our experiment, we specifically wanted to focus on this experience. How can we use this experience that they have in their current or recent context can use to enhance the resilience to environmental change? And since we are interested in sensory mechanisms, we wanted to create experiences based on social context and non-social context, but tapping onto three sensory modalities. We wanted to create social context, social experiences, or non-social experiences that can have influences on their lateral line system, visual system, and chemosensory detection. So all the experiences that we created were targeting or tapping onto these three sensory modalities. The next one. So this is how we created our sensory experiences, and we kept fish in these different sensory experiences for 6 weeks just to make sure that they have enough exposure so that we can see whether these experiences actually have any impact, positive or negative impact, on the way that they will try to keep up with the environmental change. So in the social experience, we allowed fish to see each other, like fish in the adjacent tanks, which is kind of tapping onto their visual sensory modality. And also we introduced fish water from adjacent tanks, which is a social chemical cue. And then we replaced half of the fish in tanks twice a week for 6 weeks, which is kind of like introducing all tactile, visual, and new chemical cues into these arenas. So we were doing this for 6 weeks to create a social experience, like enriched social experience. And for the next treatment, we have a non-social experience where we placed colored glass tubes in the tank. So by having differently colored glass tubes, we were trying to break the light penetration inside the tank so we can create a different visual environment. And in addition to that, we change how fast the water is flowing into these tanks time to time to create like variation in flow conditions, which is really tapping onto their lateral line system. And then we added a chemical cocktail, which didn't have any social cues, but had chemical cues related to their environmental attributes. So these changes were again done twice a week for 6 weeks to create an enriched environment that doesn't have any social experience. And for the control group, we just kept fish in their undisturbed conditions, but just to make sure that the netting behavior that was happening in the social experience didn't have any impact, we just moved a small fish net time to time in the control group. But other than that, there wasn't any differences in terms of enrichments that were done in the control group. Next. All right, so after fish went through these different sensory environments, enrichments for 6 weeks, we wanted to test how this previous experience coming from different sensory environments will impact their ability to keep up with exposure to artificial light at night. And we specifically wanted to focus on artificial light at night in the first experiment, mainly because there's a whole lot of literature that suggests that light pollution is causing many different concerns and issues for organisms at the individual level, at the cellular level, also at the community level. So some of these impacts that we all might be familiar with is that light pollution and the sky glow can affect the navigation, migratory patterns of many birds and insect species. And also we know that at an individual level, the individuals who are exposed to artificial light at night or extended light exposure, they tend to have low immunity level. They are highly likely to get infected by parasites. And also they have higher levels of glucocorticoids. And interestingly, they also seems to have impaired spatial learning and memory and disruptive hippocampal neurogenesis, which is really important in terms of performing their cognitive capabilities. So there's a lot of things that the exposure to artificial light at night can do to organisms. So we were really interested in knowing how these experiences coming from your sensory enrichment or non-sensory enrichment might ameliorate the effects of exposure to artificial light at night. The next slide, please. All right, so to create this artificial light at night exposure, we divided the fish that went through this social enrichment, non-social enrichment, and control group into 2 big groups. And half of these groups were kept in extended light exposure, where they received 17 hours of daylight and 7 hours of darkness for 6 weeks. And for the regular light exposure, they were kept at 14 to 10 light dark cycle, which is the standard light dark cycles that we keep zebrafish at. And again, they were in these conditions for 6 weeks. And then we performed a bunch of experiments to see how this light exposure affected these fish that went through different experiences. But mostly we were interested in looking at their previous experience in social context or enriched non-social context had any impact on ameliorating the adverse effects of ALAN exposure. Next slide. So these are the different experiments that we did, and each one of us are going to share the findings for these experiments. So to determine cognitive capabilities, we did a detour assay, which was developed with the help and guidance of Caitlin. And for sensitivity, we did 3 different assays to determine the sensitivity of 3 different sensory modalities. Optomotor response is focusing on the visual acuity, and tactile motor response is examining the functionality of lateral line system, and the chemosensory response is focusing on how sensitive they are to chemical cues. And then we also looked at any variation alterations in social behavior and baldness. Oh, okay, no. Emilia, do you want Kimberly to take over and do her presentation?
Yes, sorry, I'm having trouble with with remembering where we're at here. I put the other slides at the very end, Pumika, but I missed this one. We'll come back to it. So,
okay, what do you want me to do? Do you want to finish my part, or do you want to?
No, I want to go through the other people first.
Hello, my name's Kimberly, and I'm going to be going over the cognitive detour assay. So the cognitive detour assay measures cognitive flexibility based on how many fish are able to approach a stimulus fish. We measured that as well as measuring motivation to see about how long it took to approach that fish. This was used in Wallace et al. 2020's paper, as well as recommended by Dr. Caitlin, as mentioned. And it was used in previous climate studies with cichlids, mosquito fish, so it's been used, and we're using it with zebrafish. So from our results, what we got, we actually didn't see any difference in cognitive flexibility, but there was a difference seen in motivation. We were specifically looking to see if enrichment and ALAN had any effect on the cognitive performance. So what happened with the detour assay is, as you can see from this really cute little graphic here, we had the subject fish right there. There was a door that would open, and then the subject fish would go and find the stimulus fish right there. By going around this barrier, it shows problem solving in some sense, and therefore that's what we were looking for. So in figure 1, this graph up here, we see that the median time for the control and physical groups are close, while the social group stands out as taking the longest time to approach. The stress of moving the social treatment fish could have affected the motivation to approach the stimulus fish. However, further investigation would be needed to fully support this claim. In figure 2, however, we see that ALAN may have impacted the time to return to the stimulus fish, especially seen when looking at control, which is down here. It looks like some of the things got cut off, but that's all right. Control is this one right here. And then control light is going to be on this side. So as you can see, they're very different. Sorry. This one is control, and this one is control light. And as you can see, there's a big difference here. If you can't see it, if I need to send any graphs to anyone, I definitely can do that.
Kim, if you could just tell us which bars it is. There's 6 of them. Maybe you could just tell us which number.
Yeah, so 1 and 4 are control. This is control is 1, and then control light is 4. So there's a pretty big difference seen there. Control light showed faster times for returning to the stimulus than control, and physically enriched light groups showed faster return times as well. There's a possibility that physical enrichment might be mitigating adverse effects of ALAN, but it's important to see that physically enriched control groups did not demonstrate similar times, bringing into question whether physical enrichment was the reason for this difference seen. Overall, we see that socially enriched fish exhibited differences in latency to approach. And we can see this trend in the social light group as well, which is going to be the one at the very end, so number 6. And so this brings up the question as how much socializing is too much, even in social species like zebrafish.
What, how many fish did you test for each of these?
So in May, we did, in June, we did 29 per treatment group. Yeah.
And did you look at exit time, and what does latency to return mean again?
So latency to return was when the fish would, so latency to approach was how many seconds it took for the fish to go to the first one. Latency to return was how many seconds it took for the fish after leaving to come back to the fish for the first time. We were wondering if there was going to be anything seen there. And with social, we actually did see that difference.
What does that mean? Come back to the fish again?
So with a lot of fish, at least in May when we saw them, they would like leave the stimulus fish and then come back and then like leave and come back. And so we were wondering what that behavior sort of meant. And so we're not exactly sure what it could mean exactly, but it was an interesting thing that we definitely wanted to investigate further.
So the latency to approach is what's termed solving speed?
Yes.
And the latency to return after they solve, the first time they swim away, then you start timing until they come back.
Yes. So I'm pretty sure when we scored it, it was after they left this little barrier here. So there's like a little hallway where the stimulus fish was kept in. So when the subject fish left this little hallway, that's when we started timing. And as soon as it went back into the hallway within 2 centimeters of the stimulus fish, that's when we stopped the time and saw how long it took.
And also, this is backwards from how it was done in Wallace and our work. The hallway is the starting area, and
Okay.
the is at the other end so we put the reward fish where it was complete opposite.
Oh, okay. It was switched around.
Yeah. And so the hallway, we then put a door at the end of it so that we could also measure exit time, which is interesting, but was also meant sometimes because we would cover up that whole starting area that they never exited, and then we never got data on all the rest of the things.
Yeah, there were definitely a couple fish that spent their time in this little area and took a long time to come out, but there were very few that ended up not ever coming out, for us at least.
How long did you give them?
We had 5 minutes in total, and then after they reached the stimulus fish, we recorded for an additional minute after that to see their behavior. I think we were discussing maybe recording longer the next time we do these trials, because there were some behaviors that seemed to keep going that we'd like to see a little bit longer after that.
One of the ones I was thinking about, I noticed, I like your observation about this visiting and then going away and coming back. One of the ones I was thinking about was sort of total activity or exploration. And so when you say recorded, we haven't tried it ourselves. We've recorded, but I haven't gotten a student to actually follow through on like using ethogram and just taking that main square area, including where the barrier is, and measuring the amount of either distance they traveled or time they're traveling.
Yeah.
Vision. So like, which may relate back to your latency to return, like how much is exploratory and how much is more about a shoaling sort of behavior.
Definitely. And I think the exploration would be a really, really good thing to observe, because there were some that were much more likely to go and kind of check out everything around, whereas some of them were almost fixated on that stimulus fish and kept coming back. So that would be really interesting to see. I'd be interested in seeing
And
that.
overall though, I mean, I guess these guys are more motivated. We needed like 10 minutes to come out and 10 minutes to solve.
Oh, wow.
If you're, I mean, in our so Kindle has their paper out in biology letters as of a few weeks ago. The ones from the least complex environments solved the fastest. She has the most amazing video of the fish just coming out, going around and finishing.
Nice. That's awesome.
And
You
then
said Kindle? Kindle was the paper?
I'll send it to you guys.
Okay, perfect. Thank you.
And, but the others that just never even get past that barrier in
Yeah.
populations. So but I wonder if you want to add activity, the reason I also bring it up is just having a bit more time that they could spend in that area, depending, obviously it depends on the question you're asking and what you're looking for, but just something to think about.
No, definitely. I appreciate that. Sounds really interesting. I'd be interested in adding that.
What do you guys think about the social treatment taking so much longer to approach?
For me?
No, Kim, I was hoping for you too. I was hoping for some of the others. So this difference here, that the social treatment is taking so much longer to approach than the other two.
Zebrafish are highly social, right? So maybe they are like, they have enough sensory feedback from social fish. Maybe they are not as eager to approach another fish. That's what I was thinking. Is this a new arena for them? So it's just once in their lives, like the first time in their lives? Yeah.
Their first time running the trial?
Yeah, I mean, like the experience in this arena, in this like assay. So it's
Yes.
Yeah. Okay.
Yeah.
Thank you.
Yeah, of course.
But
Okay.
yeah, that's what I was thinking about the other. Maybe they are not as eager as the other fish.
Thank you.
In terms of just pure mean eye size, the social fish had much smaller relative eye diameters than the physically enriched and the control fish. So there's a chance that that led to some sort of like visual deficit that caused it to be difficult to navigate to the other fish if they were relying on their visual system.
I had another question in terms of your two parts. Let's see. In terms of your solving speed, there are different ways to consider that. Did you measure it from after they reach the barrier? Because you want to separate out your time so they're not related to each other.
So
Or
we started, sorry, did I cut you off? I'm so sorry. So when we started the timer was when we lifted the door. That's when we started recording. And then we stopped the timer when they came within 2 centimeters. However, if they were kind of just putzing around and never reached that fish, we stopped after that 5 minutes.
Well, motivation was measured in ours as the time to leave the starting area and go up to the glass.
Okay. This part, the middle part?
Attempting, if motivation to reach. And after that second, you essentially start, you have a total time of 5, let's say, but you start a new time so that motivation's unrelated to or uncorrelated to solving. And so solving then is once they've reached the barrier, it's all the rest of the time until they reach their friend.
Oh, okay. Gotcha.
Then you might have a better sense of what's going on, because if it takes most of the time just to find out, see whether they even see the other individual, I think it might clear up. Maybe it could not. But give you a better ability to understand what you're measuring, because that's a multiple levels, what you're calling approach, because we don't know when they saw it and then when they tried to find it. And that's the goal of how detour tests is. So motivation is one component of the cognition, and the other is this inhibition that's required to swim out of view of the reward fish while trying to access it. So they have to do something opposite of what they'd expect. And so that's that other component of cognition that you're measuring there.
Okay. Yeah, I'd definitely be interested in looking over the videos again and separating those.
Have fun. Sorry.
Thank you.
Take some time. And so but I do think it's worth recognizing that. So I attached in chats Kindle's paper, but then my next thing is, I'm assuming your figure 2 is log also. It says latency return unless I don't know. Because they're almost, I can barely read that. It looks like it's also 0 to 4. But I'm assuming it's log, and generally it helps to talk about this if we could talk about it in actual seconds, not log of the seconds, so I could have a better understanding of their actual, think about their behavior. And I guess you're log transforming because it's not normal distribution, or what?
Yeah, it wasn't distributing normally, so we did log, but I do have some graphs from R that were non-log. They're just not on the presentation.
Yeah, I get it, but it did make it normally distributed just by log?
Yes, I believe so.
And just again, as in terms of like a discussion and thinking about it more, for me, I mean, box plots are good, but just as even having the individual responses within, so dots for each individual for our discussion could help to get a sense, so that I really know feel like I know what your fish are doing.
Yeah.
Okay, I'm going to keep us moving so we can get through the rest. No, that's great. Wonderful, wonderful input. Kim, maybe you can write all that down while we're moving to the next person. Thank you. So we're going to move to Sean. These aren't quite in the same order that Pumika brought them in, but I think we'll make it work.
So I was in charge of the boldness assay, specifically the social boldness, and examining the behavior when exposed to a novel stimulus object. So we can see the 3 bar graphs here. All the way on the left, we have the control enrichment. In the middle, we have the physical enrichment. And on the right, we have the social enrichment. So as you can see, our control group, although it's the most varied, it also has the highest mean by far compared to both the physical and the social group. Although the difference is not technically statistically significant with a p-value of 0.06, really, really close. We're right there. So it's pretty likely that we can say that there's probably some effects from physical and social enrichments in terms of the zebrafish's social behavioral responses. So that's sort of my main takeaway for this slide, is that there very likely is something going on, but we can't statistically say, yes, there is. Next up will be the ALAN exposure, or the artificial light at night. So this one's a little bit less clean cut. First off, we're going to start by comparing the first and the second chart, or the first and the second box plot. So the first group is our control group. The second group is our control light exposed. And as you can see here, our control light exposed takes twice as long, basically, to react to that novel stimulus object when we're comparing the mean. So again, we can't say not quite statistically significant with a p-value of 0.36, but we can say, hey, there's something going on where it seems like our light exposed fish are taking longer. To continue on this point, we're going to compare our control light exposed to our physical light exposed. Again, we notice the enrichment conditions usually lowered our response time. Once again, they do here when we're comparing that second chart to the third, or the second to the third box plot, we can see that our control light exposed takes longer than our physical light exposed, showing that the physical enrichment probably ameliorates some of those ALAN exposure effects. And then finally, we're going to compare that second chart to the fourth chart, and we're going to see that our control light exposure and our social light exposure have very little difference between them. So there could be something going on, but again, with our p-value very, very high, 0.68, probably not something that we can really take away. So in terms of this, we can say, hey, maybe there is something going on with these enrichments that is sort of ameliorating or helping these fish with the light exposure, but we can't really say statistically that something is absolutely going on. So that's my piece. Any questions?
Can you remind me how did you do the boldness assay? I think I
So
lost
for the
it.
boldness assay, we had them in a 10-gallon tank with nothing in it, and we lowered a bead on a string from above, and then as soon as it touched the water line and the fish were exposed, we started timing and timed the latency to expose, or latency to approach, excuse me.
So does that mean it's more like it's also neophobia?
It could be. So like with the social group, that was a little bit of my hypothesis, is that because these fish are constantly having, new tank mates, they're being exposed to these new conditions, they might be a little bit, more prone and adaptive to something new being dropped into their tank compared to the control. But then again, with the physical group, they're only having the lateral line stimulation with the increased water flow and a little bit of chemical cocktail. So there could be a difference. I don't know for sure that is what I would blame it on, but it's something to do likely with the social and physical enrichments that we had at play. So some factor that we changed.
So I'm seeing possibly an effect of the enrichment, but not much effect of the ALAN.
I wasn't really wanting to say that there was too much effect from the ALAN. The first graph, I really am comfortable saying, hey, there's likely some takeaways here. I don't know that there's anything very clean cut with the ALAN exposure.
Well, I think that's still a result.
It is. It absolutely is.
That we're not getting much of an effect of ALAN. It's something to discuss later if we should continue with the ALAN or if we should go the other way and do less light.
But at least I think the trend from the control light and social light, you have like this, I mean, they are more like alike, right? So because I am thinking about the bigger question about if social enrichment actually can help them deal with climate change. In this case, the ALAN effect. And these two groups are actually more closer together. I mean, I know that it's not significant, but at least.
But they are performing better, yes.
Yes.
Well, from the p-values, these two are both these are more similar to each other. So I think we need to look at these data, the individual data points a little bit more, like what Caitlin was suggesting, before saying much of anything here.
Mm-hmm.
But it's a good point. I think the social here, the similarity between the social and the physical enrichment is really interesting because as Sean was pointing out, there's no constant change in the physical enrichment setting, right? There is a constant change in the social setting. And so it's something else that makes them similar to each other.
But the physical is also sensory, right? Like the lateral line and
They get the water stimulation with the increased flow rate and the chemical cocktail.
Yeah, but the chemical is the only thing that, well, was the water flow changing?
Yes, we would increase it. It was 2 minutes, right? For the physical 2 minutes, we would increase the water flow rate in their tanks and then lower it back to the normal parameter.
Yeah, so a little bit more shift. Okay, let's keep going because I'm now a little worried about getting through everybody. But thank you.
Thank you.
So I focused mainly on the optomotor response test, and I apologize for not having any graphs on here. First of all, I had an 8-slide presentation with a lot of graphs, and it just didn't fit on here. But if anyone is curious, I'd be happy to send those graphs out. But for the optomotor response, basically we're testing the visual acuity of these zebrafish, and to evoke the OMR, we placed the fish in a rotating cylinder with a striped pattern. And there was 6 of these patterns ranging in various black and white bar sizes. And how we scored it was based on the resulting swimming movements after the apparatus was turning clockwise for about 30 seconds. That's how long we recorded it for. We scored it based on either one, the positive, if it was a positive response and the fish did 2 consecutive circles in the right direction, or 0 if the fish did not respond to the visual cue. And so we had 2 sets of data by the end of the summer experiments, which was then compared. There was no significance found based on the data analysis for any of the experiments. All the p-values ranged from about 0.52 to 0.86. But I think 2 interesting things that I found just from looking at the graphs was when looking at the ALAN experiments, the control performed better. The control light, performed better on most gradings in comparison to the control group. And then in relation to that, the social light performed better on most gradings in comparison to the social control. But I should also say the social overall performed the worst amongst the 3 different enrichment groups. So that was something that was unexpected that we found. But of course, there was no statistical significance behind it.
So again, the artificial light at night is improving their response. Is that right?
Yes.
I feel like we should keep going to the eye size before we discuss too much. Is that okay?
Yeah.
Okay. So for the eye size, there was a bit of significance. I just put the 2 charts that indicated that here. So when we were just looking at ALAN exposure for the control versus light exposure, we didn't see any significant difference in relative eye diameter. Now, when we added physical enrichment into that concoction, we saw, once again, a similar lack of change. However, when we focused in on social, we had a very significant drop in relative eye diameter. These were all relative measurements, but I know that Amelia and I were in discussion, and there's a possibility that it is still due to body size that we're getting this big skew in results. So there's no really clear indication that it's caused by ALAN, despite the significance of the p-values, which I'll provide in just a moment. But when we were just looking at enrichment type, we can see the control had a fairly large relative eye diameter amidst the individuals within that. And then from there, physical was fairly comparable. And then when we went into social, we see a significant drop. The p-value for this was 5.1 times 10 to the negative 6th. So it was a very, very significant value. But we're uncertain if that was due to like just a general attribute of size or if that was a meaningful result. And then when we focused on enrichment type with ALAN, specifically with the social enrichment type and ALAN exposure, we saw that the socially enriched fish, once exposed to ALAN within the females and males, both experienced smaller relative eye diameters. So this is still like this significance value was 9 times 10 to the negative 6th. So both very significant, but once again, subject to the same uncertainties. But yeah, it still does align with what Vaishnavi had found for hers, but we're not really certain as to what promoted this trend.
Yeah, let me go back to say that I think it's actually quite meaningful, the body size result. I'm sorry I'm losing my voice here. I hope you can still hear me.
Plenty of
the body size result is that the socially enriched fish are considerably smaller than the other ones, right? So they have smaller eyes, but in part because they are just smaller. And that may be something that impacts all of our results across the assays.
Most definitely.
Isn't there a way to correct by size?
So all of these results were corrected by size, but even then, I guess there's, in the sense, like I did the eye diameter divided by the standard length, so they should be per like relative values. But I'm not certain that it still had an impact. Sorry, Amelia, I cut you off.
So I might have missed, I probably missed it, but were the fish were reared since egg, or how long? Why would eye size change in their lifetime?
So they're, I'm sorry, go ahead.
Oh, no, it's all right.
The treatment was 6 weeks, right, in each treatment, so that they were adults when they went into it.
Yeah.
But they could be smaller overall because they're eating less, right? So they would have grown over that time somewhat.
And you mentioned their size being different, then that made me wonder if you controlled for size in the cognition studies, because we keep that in there as a covariate, because obviously it can affect the speed they swim. So something else.
It's true of everything, and we did not put body size into any of the other analyses.
Yeah.
Right.
Amelia, I did have a quick question. So if we did use relative measures, do you still feel that general body size would have a big impact? And if so, is there a way to correct for that?
Yeah. I think we just need to look at the relationship between body size and eye diameter.
Why I was asking
to see if a linear correction, what you did was just to divide by it, and to see if that's appropriate. We need to look at that relationship. But regardless, I just find the finding about the body size very compelling. Actually more compelling than I do about the eye diameter. And it may be that the eye diameter is even smaller in the social ones. But I'm not quite sure what that means.
Okay. And then to respond, I guess, to a previous question about eye size changing over time, I think one thing that Vaishnavi and I considered in the previous presentation was the fact that ocular pressure could lead to changes in general eye size. And so if they're being stressed because of a greater exposure to ALAN, there's a chance that their eyes could have expanded as a result of that. In that instance, a larger eye diameter would probably be more indicative of a stressful environment and cause visual deficits if we're using eye size as a proxy for visual acuity.
Yeah, it's also something we didn't think about doing the stress hormones on these enrichments, right? And we may want to rethink that going forward. I don't know why it never occurred to me that the social treatment would be particularly stressful, but it might be. Shall we go forward?
Sounds lovely.
We still have a couple more.
So I'm going to be introducing the rheotaxis assay, rheotaxis being animal movement in response to water flow. And so we use our tactile motor response to test the sensory resilience and their reliance on tactile cues. And so we expose them to 4 different speeds in a swim tunnel. The 4 speeds being 2, 4, 8, and 10 centimeters per second. They were exposed to the speeds for 30 seconds, and then there was a reduction in between period for 30 seconds before they were exposed to the next speed for another 30 seconds. And I have this video here. It's showing positive rheotaxis in that the fish is facing the direction of, or it's facing the stream. And yeah, so sometimes it gets pushed back, but at about it'll slow down and it'll show like it not reotaxing once it slows down. So we exposed them to different speeds and looked at if they were reotaxing or not. Yeah. Oh, not yet. I think, yeah, this is when it slowed down and then the fish has stopped reotaxing. It's going in all these different directions. It's not as strong.
Okay. Alyssa, you haven't started scoring, right?
No, I have not.
Yeah, so this is something that we're looking forward to in the coming months. We'll find out if they had an impact. Just wanted to get all of the assays up here. Thank you.
Thank you.
And I think, Pumika, we're going back to yours now.
And Emilia, I wanted to add that the smaller body sizes that we see in the social assay could be because that we swap like 50% of fish like twice a week. So they must be like fighting and like engaging in like reestablishing their social hierarchy. So they might be like spending a lot of time like aggressive interactions, which are highly costly in terms of physiologically. So maybe that's a possible explanation for that.
Yeah, for sure.
Yeah, so I am going to be talking about the chemical motor response CMR, which is actually the piece that Jasmine Norris completed. She's not here with us this semester, but this is the piece that she was leading. And in this part of the essay, we are going to be looking at how sensitive zebrafish are to chemical cues. And this is the setup that we use, like a modified version of this. The fish are kept in a compartment and a chemical cue is presented to the fish, and then we'll look at whether they approach the side of the compartment that the chemical cue was presented. And we used alanine as our chemical cue, which is amino acid that gives out the smell of food. So it's a chemical cue in the in foraging context. And we used 4 different concentrations of alanine, presented these concentrations at a random order, and we looked at within 30 seconds whether the fish approached it. So it's very similar to what we were doing with OMR and the rheotaxis assay. We always looked at for 30 seconds, we give them 30 seconds and look at whether they respond, display the expected response to say whether they responded to it or not. So first, I want to show you what we found in terms of the exposure to ALAN and how it affected the CMR response. So in here, we are looking at the 4 alanine concentrations. A1 is the weakest, A4 is the strongest concentration we had, and we are looking at the percent response on the y-axis. And the blue is for the control, and orange is for orange or brown. For the ALAN group. So what we see is, which is very similar to what Vaishnavi found, when they're exposed to ALAN, they actually had a better response to chemical cues compared to the control group. And yeah, that's not what we were expecting, but they did. I mean, there isn't a statistical significant difference, but still, if you just look at the pattern, they did better than the control fish. And then we wanted to look at whether the previous experience in sensory or non-sensory environments had an impact on the way they respond to these chemical cues once they went through the ALAN exposure. So here we're looking at the blue is for the control, which means that the fish that experience ALAN treatment, just the ALAN, And green is for the fish that went through ALAN, but before that they went through the social experience. And the orange is for fish that experience ALAN, but before that they went through the non-social or physical experience. So again, seems like data is all over the place. We didn't see a significant difference, but still we see that the fish that experience ALAN exposure, just that, with no sensory enrichment, responded better to these chemical cues than the fish that experience enrichments. How we are gonna explain these results, it's little complicated. We're still trying to figure out what could be the possible explanation. But when I was scoring the videos, when I was looking at the videos, it seems like the stress that was that fish were exposed to during the assay was really like had a role to play in it. But we are coming up with ways to make the setup more smooth and less stressful for fish. And we've done this experiment before with only 3 alanine concentration, and this is the first time we did with 4 different concentrations. So yeah, there are a lot of things that we need to be thinking about in terms of continuing this assay in the winter.
Mika, can you speak to why is this a flat pattern instead of a down? And which one's the strongest concentration?
A4. Yeah, it's really unexpected. Like,
and which are the ones that you used before? Or
A1 through A3.
Because before you had a down pattern, but this pattern's reversed, is it not?
Yeah,
it should be the other way.
Yeah, I think I think it's the stress. Like, I looked at how many times we did the A4, I think, as the last, and then there were more opportunities. Like in more trials, A1 was last, so maybe that's contributing. That's why I was saying that when they went through like 4 different exposures, it was kind of building up the stress level, and they were like not responding to it. Like they were not leaving the compartment just to like respond to it.
Right.
And there were like issues with how fast we removed the divider. So by the time they removed the divider, I feel like that the chemical cue was like dispersed all over the arena. So I could clearly see that the water is moving. So those are the things that we need to really like touch on in terms of like refining our protocol.
Anybody else want to jump in? Should we keep going?
To see where the water's going, just some dye that you put in, just like you put the alanine in.
Yes, we that's how we started with like determining how fast we need to like press the syringes and everything. But when you do multiple people, like with the personal like differences, I could see like, yes, we didn't use a dye because we didn't want to have any visual impact when we are running the assay. But when we were doing pilots, we did use dyes and to see the movement of the chemical cue. Yeah.
With or without the fish in there?
For the pilots, we first did without fish because we wanted to see how the chemical cue is like getting transmitted across the test arena. And then we tested it with the different concentrations to identify which concentrations that they display the 0 response and which concentration they display 100% response and 50% response. That's how we came up with the 3 different concentrations.
But once they're swimming in there, the movement of the concentrations, like what you saw with the dye, changes when the fish is moving.
Well, it's a small test arena. I mean, we cannot control for everything. So what we try to do is we just like the movement of fish is like, I would say it's like 15 centimeter, and the purpose was to like let it flow through the arena. And then the fish will detect it even when they're at the compartment that we were holding it, and then they respond to it, like go to that side of the arena.
Okay.
Yeah.
And so in terms of so you said you're thinking about maybe changing your social versus non-social environment. What was the density of fish that you were using?
We had 12 fish in 8-liter tank. So we didn't do the calculation, but it
Well,
was
that's enough.
constant for all groups.
And the non-social was
It's the same number of fish. in the
just
same
no
tank.
added fish into it.
Yeah, we scooped out 50% of fish and we added new, like, I think 6. Like we moved 6 out, we added 6 from the adjacent tank. So it's always maintained at the same density.
Oh, no, I get that, but only in the social one, or?
Only the social fish group had the swapping event taking place.
So it might help to like relabel like what we think this is, because if their densities are the same, their social component is the same, and it's more that like all our thoughts could change if we said something like, gene flow, or, I'm trying to think of, or new social environment versus same social environment, or dear enemy, or, I don't know which term would be the right one, but I think that would help our interpretations, because saying social and non-social, sort of, you guys get it, but
we don't even, like, we have disagreements. Caitlin, we have disagreements. We've been talking about it. So if you have any suggestions, that would be great, because I know when we started it, we used to call it social versus physical experience, but then what we are trying to do in the physical environment is not to give any social cue, because we are adding a chemical cocktail, which is not really like the standard physical attributes that we talk about when we talk about environmental features. So if you have suggestions, like, we are all ears.
Yeah, so it may spend a little more time, as I made a couple suggestions on how you define the environments. That might be the starting point. And even if you just use more.
True, the non-social group.
I think we lost you, Monse. Yeah.
Did we lose Monse?
I think we.
I'm sorry, my connection.
You're back.
Fails all the time. I'm sorry. I was going to say that the non-social group also has social like they are experimenting other social fish, right? And they are also experimenting the chemical and the visual. Maybe in a little, like, they have differences, like small differences, but they are also experiencing those sensory stimulants.
I think the initial idea was that they would be enrichment, so it's social enrichment. It's not just that it's social, it's that it's being enriched by the social group being switched out periodically. Right.
The term
I'm thinking
is usually
they are forming hierarchies. Sorry, Caitlin, sorry.
Yeah, no, I'm just gonna wait.
Go ahead. Sorry. No, I was just going to say that they are forming also hierarchies.
Yeah, that's why we were thinking like in control and
the social group. So like Pumika was saying.
Yeah. Okay.
Yes, go ahead.
I
don't know if my connection fails.
I don't want to run out of time here, so let's keep moving just to the December experiment.
So for the next step for us is going to be repeating the same experiment, same set of experiments during the winter break. But this time we will be looking at how they respond to elevated temperature. So we are only changing the climate factor that we're going to be investigating, but we will repeat the same sensory experience, but hopefully with better terminology to explain it. But we will put them through the sensory experience for 6 weeks, and then they will go to the climate conditions, and this time it will be elevated temperature. Another 6 weeks in there, and then we will start our experiments. And we wanted to look at the elevated temperature because we already know that increased temperature are known to affect like multiple different sensory modalities, like they disrupt and damage the sensory receptors in tactile. That's important for tactile sensation and also chemosensory detection. And also we know that as the temperature increases in the environment, especially in aquatic systems, the surface water levels increase temperature. And when that happens, many organisms tend to go to the deeper water levels, which creates like which expose them to completely new environments. It could be different in terms of their visual environment. These environments, the deeper waters could be darker. And also they might be experienced in different communities that they are not familiar with, like different predators, different heterospecifics to interact with, to compete for resources. So again, these can have multiple different impacts when the temperature increases in their natural habitat. So we're going to be looking at how this elevated temperature will affect the behavioral responses, and also hopefully can we use the sensory experiences enrichments to ameliorate any of these adverse effects that we might encounter due to the elevated temperature. And I think it's pretty much what I have in the next few slides, Amelia, if you go to them. So
yeah, let me go back to one. No, that's it. Sorry. That is what you had. Okay. So I wanted to raise a question, just one last thing before we close up here, which is that in between those two, we're also going to, PMIC is going to generate some fish for Christina's transcriptome experiment and for DeLawrence's colitis work, right? That was the original plan. PMIC, are you there? Can we get her back for a second? So the question I wanted to raise was whether we should, is about the ALAN in particular. So with these two experiments, the idea is to use multiple environmental factors, right?
Yes.
Maybe you could just state it really fast.
So in the AM1, we are going to be looking at 4 different climate factors: elevated temperature, acidity, flow fluctuations, and ALAN exposure. And then we wanted to combine all these different possible combinations that we can come up with, like to see which interaction, because when you talk about the environment, one change in environmental factor is not how it's happening. Everything happens at the same time. So the interaction between these multiple environmental factors, how is it going to be affecting an organism's ability to display resilience? So we're going to be looking at all these possible combinations, and we are going to expose zebrafish for these different combinations. And then the original plan is to run behavioral assays, physiological cognitive assays on these organisms. But then Christina will come in and also looked at the differences in transcriptome expression when they are exposed to these different combinations and different climate factors.
Yeah, can you throw DeLawrence's in there too?
For DeLawrence part, DeLawrence, do you want to talk about the colitis one, or Emilia, what?
No, I'm sorry. It's just because my voice is going here. I was hoping you could explain the treatments on DeLawrence's.
Okay, so in DeLawrence part, where he's looking at the impact of colitis exposure, we are putting zebrafish through just the enrichments for DeLawrence. So we have the control group, we have the sensory enriched group, the socially enriched group, non-socially or physically enriched group. And again, they're going to be going through the same exposure for 6 weeks. And then we are going to ship fish to DeLawrence's lab. And then he's going to be exposing them to colitis. And if I remember, he's going to do 2 injections. And then he's going to do the dissections to look at the infection rates and how the previous experience in these sensory enrichments will ameliorate, make it more adverse. Like we don't know what the pattern is going to be, but we're going to be looking at how we can explain these previous experiences in terms of their infection rates.
So it's the same as this summer in the sense that fish are going to go through these enrichments for DeLawrence's piece, and then they're going to go through the climate treatments, no enrichment for the transcriptome and Christina's piece, right? So I just wanted to be absolutely sure before we did that we want to do everything as we have in the past. And the one thing that's occurring to me, looking over the preliminary results, it looks like maybe we're okay. But the one thing that I think maybe we should shift is the ALAN. And this is because, so in the grant proposal, we actually wrote it the other way around. It was going to be less light, right? To match with some experiments that Piyomika has done in the past that show that there's an OMR difference with less light. But we wanted to try ALAN because it seemed to make more sense from a climate change perspective. But I'm not seeing a result here that we can use, right? I'm a little worried about that result. So I'm thinking maybe we should switch the ALAN to less light. And just wanted to get your input real fast on that. Any?
Less light could make me think of more turbidity. And that I know, at least for zebrafish, is relevant, I think, where they're found in the wild from reading.
Yeah, that's what we were thinking, is sort of that also if the temperature goes up, that they may go down in the water column, right? And it might be cooler down below, darker down below, right?
In doing ALAN, one of the things, so we've also published on that really I'm constantly concerned with is what level is being picked. Just because we call it ALAN, you know, I see how many hours of light they got extra, but not how, if it's brightness that's extra. And that's the big thing. And so maybe that's the difference. Not so go for 12 hours, go for both treatments 12 hours, whatever amount of light, but one is dimmer than the other. And that really, I think, would get a lot at what you're saying. I mean, of course, environment is important, and I'm not diminishing it. We have not found much going on with fish with gambusia, but that was just one experiment and one thing, and I get that. But I would think that if you want, if by dimming it, you could relate it to either going further down in the water column or more turbidity. And then, since we're already seeing what you said.
Yeah. Any objections to that? I think Piyomika and I will just argue it through and come up with a decision. But whatever we do, I think we have to keep doing moving forward, right? Okay. Well, if you have any thoughts about that, get back to us. Let's continue this conversation.
Could you remind me when you're taking hormones? Because I mean, I know you've said you should have here and there or could here and there, but at this point, now that I've heard it, if and when you plan to take hormones, I'm going to say why I ask, which is a lot of times it's earlier on in the stimulus that their response shows up and then they habituate. And so we just want it, depends on what our question is as to when you want to take the hormones.
Yeah, so the plan was to do the hormone experiment next summer. Yeah. So we need to think through those details, the specifics of how to do it. But my thought was we could wait until January and then get together again and maybe make a decision about specifically how we're going to do it at that point. Is that okay?
Yeah, I just wanted to not let it hang if it's going to come up for earlier.
Piyomika, I can't see you anymore. Are you okay with this too?
Yes. Yes.
Okay. Good. Let's wrap up then. I'm sorry to keep you all a little late than we expected. But thank you for your input, and let's keep the conversation going. Like I said, we can do email for a while. Thank you all for presenting too. Really appreciate it.
Can I offer for Kim to get in touch with my grad student Kindle, as I think it's always productive for students to interact with each other and learn from each other? Tyndall has struggled through public this paper and learned. So who can I send an email to, or can I just put it in the chat?
I can. I was going to say that I could get your email from possibly Amelia and send you an email with my email.
Yep, do that. Okay, told you, but I
Sounds
think
good.
Sounds good. And just in general, I'm happy to give people emails if within the group to trade around. So if you need to get in touch with anybody, feel free to contact me. Okay, thank you, everybody. I'm going to close the recording, and we'll see you again soon, hopefully.
Great.